Alright, Waxahachie Global, we're going to finish up this chapter with Concept 6.6 and 6.7. 6.6 is all about the cytoskeleton, which is basically the structural framework that holds the cell together. Scientists used to think that these organelles that we've been talking about simply floated free in this cytosol, that there was nothing that kind of bound them to one place, and that there was very little structure inside the cell, that it was basically just a bag of water. But now we know that's not really correct, that there is structure inside the cell. There's something called the cytoskeleton, and it's a network of different types of fibers that organize structures and activities inside the cell and provide a framework, sort of like a skeletal system for the cell inside. The cytoskeleton is made up of mainly three different types of structures. One of those is called the microtubules, and those are the larger of the structures. If we're still using our comparison to a cell to a city, microtubules would be like the largest concrete pillars that hold up, say, a football stadium or any extremely large structure. So these are the largest ones, microtubules. Next in line is the microfilaments. The microfilaments are actually the smallest of the three, and they would be more like cables. So think of like a suspension bridge and the cables and wires that hold up a bridge or something like that. So microfilaments are the thinner of these three, and they're more like cables or chains. And then last but not least, we have ones that fall in between. These are called the intermediate filaments, and of course, based on their name, intermediate, you can probably guess that they are somewhere in the middle in size between the microtubules and the microfilaments. Here we have a great photograph taken through a microscope of a fluorescently dyed cell with the cytoskeleton showing. The green lines that you see represent the microtubules. So these are all the larger of the structures we're going to be talking about. The red that seems to be kind of around the outside, you'll find out those are the microfilaments. Microfilaments tend to be gathered right under the cell membrane, so you'll see they form this material right under the cell membrane. And then we have the blue, which is the DNA of the nucleus. What we don't see in this particular shot are the intermediate filaments, but we'll discuss those and we'll see a picture in just a little while. So what is the purpose or the role of the cytoskeleton? You could probably guess that primarily it's support. The cytoskeleton helps support the cell and allow it to keep its shape. It's also involved in motility, which is another word for motion. Just like your skeleton provides support for your body and provides a place for muscles to attach so that you can move around, well, this cytoskeleton also provides those things for the cell. It provides shape and support, and it allows basically for motion within the cell. So there are types of proteins called motor proteins, and those would be at the cellular level similar to like your muscles at the entire organism level. And so these cytoskeleton structures, they serve to help give the cell its shape, but they also serve kind of as a highway system. You'll find that these vesicles that pinch off of the Golgi apparatus or the vesicles that pinch off of the endoplasmic reticulum, they actually use the cytoskeleton as a monorail or like a train track to travel from one part of the cell to the next, and we'll talk a little bit about that. Also, recently we found out that the cytoskeleton may play a part partially in certain biochemical activities of the cell, so it may not just be structure, it may also play a part in the biochemical activities as far as when certain things occur or how they occur. The cytoskeleton is especially important in animal cells because animal cells don't have that rigid cell wall, so they need this cytoskeleton for support and structure. Here's an example of the cytoskeleton being used kind of as a pathway or a train track or a monorail track. At the top you have a picture, or a diagram I should say, a drawing of one of these vesicles. And this vesicle is kind of a transportation vesicle. Maybe it's transporting proteins or some other material that was created inside the Golgi apparatus or inside the ER. And it uses a motor protein that basically walks along this microtubule. So the microtubule is kind of like the pathway, and the motor protein moves that vesicle along as if it were a railroad car on a track. Down below we have an actual microscope picture. Here is the microtubule, you can see there, and this is the vesicle, and it's traveling along that microtubule. We've already mentioned the three components, but just to clarify, They are the microtubules, which are the largest of the three. Then we have the microfilaments, which there's another name for those called actin filaments. They're made of a protein called actin, A-C-T-I-N. They're actually the thinnest of the ones we're going to look at. And then the intermediate filaments are fibers that are somewhere in the middle between those two. We're going to spend most of our time talking about the microtubules and the microfilaments, and just a brief amount of time talking about the intermediate filaments. Your book does a great job of kind of summarizing all three of these. So if you look on page 113 of the textbook, you'll see a nice summary with pictures and tells you the structure and the main function of each of these units or each of these structures. So that's a great resource for you, textbook page 113. We'll take a look at each one just briefly, so you'll have it here as well in case you don't have the textbook with you. So here's the first one we're going to look at. These are microtubules. Again, here is a fluorescently dyed photograph taken through a microscope. The blue represents the nucleus and DNA, but the green is the microtubules that run throughout the cell. The microtubules are made up of a material called tubulin. They're hollow columns, and you can see it's formed from this tubulin molecule that kind of spirals to shape this pipe or tube. They are mainly for cell structure and shape. Consider them like the steel girders of a building. They resist compression, so they keep the cell from getting squashed out of its shape that it needs to be in. They're also very important for motion in the cell. We're going to talk about how microtubules make up either the flagella, which is kind of like the little tail that you sometimes see on a cell, or they also are involved in creating the cilia. The cilia is kind of like the little hairs that you see that allow for motion of certain microscopic creatures or cells. They're also involved in moving chromosomes around when cells begin to divide, and they also can move organelles around throughout the cell. Here are the thinnest and smallest of the structures we're going to look at, called the microfilaments. They're also called actin filaments because they're made out of a protein called actin. It's basically a strand of two intertwined actin polymers. So it kind of reminds me of a rope or a chain that's made up of two fibers that are intertwined together. These are also about maintaining cell shape. You find a lot of the microfilaments right directly underneath the cell membrane. They are more like tension-bearing elements, so instead of resisting crushing forces, they kind of resist pulling forces, and they can be thought of kind of like cables in a suspension bridge. They're also very important in muscle contraction. That's probably where you've heard of them before. Actin along with myosin, we'll talk about that. They're also important in other types of movement, such as cytoplasmic streaming, which we'll talk about, and movement for creatures such as amoeba and other pseudopodia, other small microscopic creatures that move in certain ways, like amoeba, the way they flow. We'll talk about that in just a little bit. They also have a small part in cell division where they form the cleavage, where that's the midpoint at which the cell is going to divide. Last but not least, we'll talk about these intermediate filaments, which are midway in size between the other two. Great shot here, again, using fluorescent dye. The yellow-orange would be the nucleus of the cell, and the green lines you see here would be the intermediate filaments. Again, just like the other two, they have something to do with maintaining cell shape. They also help anchor the organelles into a particular spot within the cell, and they also are responsible for the formation of this nuclear lamina. The nuclear lamina, remember, is kind of part of the whole nuclear membrane that separates the nucleus from the rest of the cell. So let's start with the largest ones, the microtubules. They are hollow rods formed of that tubulin material that we talked about. Again, they're primarily kind of as the internal skeleton of the cell. They hold the cell's shape, they withstand compression, they help the organelles move around throughout the cell, and they also separate chromosomes during cell division. All eukaryotic cells have microtubules, so you would find these in plants, animals, dogs, cats, zebra, you name it. In animal cells, an important part of the cell is usually near the nucleus. It's called the centrosome. The centrosome is an area near the nucleus where the microtubules grow from, and it's where they are organized. So in that centrosome area, you have usually a pair of what's called centrioles. And centrioles are a structure that basically is where the tubes, the microtubules, grow from. The interesting thing is that not all eukaryotic cells have centrosomes and centrioles. Animal cells do, but most fungi and plant cells do not. So it doesn't seem like you have to have these structures to have microtubules, because fungi and plant cells have them as well, but they are an important part of animal cells. And again, they come into play, especially when the cell is beginning to divide. You'll see the chromosomes line up in the center of the cell, and that has a lot to do with these microtubules, centrosomes, and centrioles. Here's a great picture taken from your textbook. At the top is a model or a drawing, and then down below is an actual microscope picture of this whole area where the microtubules are being organized and built would be called the centrosome. And then within that, there's generally two centrioles, and you see there are these structures made of microtubules. The picture or the drawing at the top kind of mimics the actual microscope view down below. We said the microtubules have a lot to do with movement within the cell, and usually cells, if they are motile, they move around in two different ways. They either use what's called cilia or flagella, and microtubules are actually the structures that build both this cilia and this flagella. The cilia are kind of like the very tiny hairs that you see sometimes on certain microscopic creatures like parameciums. So cilia tend to be quite numerous, and they cover the entire outside of the cell or the microorganism. Flagella tend to be like what you would see, say, on a human sperm cell, where they seem to have a single whip-like tail. Sometimes you'll see two or three flagella, but oftentimes you only see the one. And this is usually larger, and it looks more like a tail or a whip as it moves back and forth. The flagella, the way it moves, it kind of undulates left and right or up and down, back and forth, and it kind of drives the cell in the same direction as the axis of the flagellum, whereas the cilia are more like boat oars, where they kind of reach out and then push back, and they kind of move perpendicular to the direction of the motion. So you have two different methods of movement, but both of these types of movement items, the cilia and the flagella, are both made from microtubules. Here's a video showing a little creature with a flagella. You can see the main body in the green, but if you watch the video carefully, click on it and hit play, you should be able to see like almost a clear whip-like tail that comes out and kind of thrashes around, and that would be the flagella. And then on the paramecium, all around the outside edge of the paramecium, you'll see these tiny little hairs called cilia, or they look like hairs, called cilia, and you'll notice how they kind of wave back and forth like oars in a rowboat, and they can move the paramecium throughout the water. Another comparison here of the motion of flagella and cilia. So here we have like what looks like a human sperm cell, or it could be another type of sperm cell, has one long flagellum, and as it moves, undulates back and forth, it moves the cell forward. And then here we have a cell covered with cilia. Oftentimes the cilia can be quite numerous, and these move more kind of like boat oars, where they have a stroke and recovery, and that's how they move the creature or the cell through the water. Both cilia and flagella are made up of these microtubules that we talked about, and they're formed in what's called a 9-2 arrangement. They have on the outside 9 pairs of microtubules in a ring formation around 2 microtubules in the center. We'll take a look at that in just a second. They're attached to the main part of the cell by what's called a basal body, and the protein that actually moves this whole structure is called dynin. Dynin is a large motor protein that can actually change its shape. It uses ATP molecules for energy to change its shape, and as it changes its shape, it's attached to these microtubules and causes them to bend one way and then the other, which allows for the flagella or the cilia to move. Here's a quick little animation. You can hit right click and hit play if you want to see this animation. It kind of shows you how this dynin protein is attached to the microtubules and how it bends the microtubules one way and then the other. Another great couple of pictures showing the microtubule structure. You see the 9 pair right here. You see the 9 pair microtubules in a circular formation with the center 2 in the middle, and then you see connection This is called the basal body right here. This would be like the flagella, and this is where it connects to the cell right there. So that connection is called the basal body. So good picture here showing all these parts and how they are put together. The way this dynein protein works is that it kind of actually walks along the microtubules. And so the dynein kind of reaches out and grabs the microtubule and pulls itself along and then releases. And then grabs, moves, and releases. And by doing that, it exerts a force on these microtubules that causes them to bend one direction and then the other. So here you have kind of like a little animation where in the middle you see this dynein proteins, and they are linked to the two microtubule couples here. And then the ATP allows that microtubule to kind of walk along one set of the microtubules and bends them toward one direction and then releases, and it can bend back the other way. So you'll have a motion to the left and then back to the right and then back to the left again. And that whip-like wave motion is what allows the cell to move throughout the water or move throughout its environment. Going to the smallest of these structures, the microfilaments. Remember, we also call these actin filaments. Microfilaments are small, twisted double chains of a material called actin, which is a globular protein. Microfilaments seem to be present in all eukaryotic cells, just like the microtubules, and they are for holding the cell shape, withstanding tension and pulling forces, and they have a lot to do with motion and movement, particularly in muscle cells and other areas of the cells. You see a lot of microfilaments directly under the plasma membrane of a cell. It forms what's called a cortex, and that helps support the cell shape. And then you also see these in a variety of places. We mentioned muscle cells, but if you'll remember, like in 7th grade science, when you studied the digestive system, you might have studied in the small intestine, there's these little finger-like projections called villi. And on those finger-like projections are even smaller projections called microvilli. Well, the microvilli are actually made up of these microfilaments that we're talking about. So let's imagine that you're looking at a small intestine, a very close-up picture of the small intestine, and it's covered in thousands of these microvilli. And these microvilli are actually made up of these microfilament fibers. And then down below, we even have some of the larger intermediate filaments that we're going to talk about in just a minute. The microfilament fibers that work for motion and movement, they work together with another type of protein called myosin. And you've probably heard of these two, actin and myosin, especially having to do with muscles and muscle cells. In the muscle cells, what you have is thousands of these actin filaments that are arranged parallel to one another. And interspersed with these actin filaments are these larger, thicker filaments of this protein called myosin. And the book says the thicker filaments are interdigitate within the thinner actin fibers. I thought that was a kind of funny word, interdigitate. Basically means they're interlocking like fingers of two clasped hands. So you have these parallel sheets of actin, and interdigitate with that are these filaments of myosin. And the way that the muscles work is when they get the electrical signal from the nervous system, these two types of fibers actually link and ratchet together to shorten the muscle fiber, and that's like a contraction. So if you were to pose in front of the mirror, and like when you were a little kid and your mom or dad said, make a muscle, and you were to flex your bicep muscle, well, you are shortening that muscle fiber and making it bunch up together. And the way that works is the actin and myosin link together and ratchet those fibers to create to make them shorter. And then when you relax the muscle, they ratchet the other direction to lengthen those fibers and to allow your muscle to relax. Here's a great model of how that works. Up here at the top we have an actual microscope picture of these fibers, the actin and myosin filaments. And then here you can see the actin is represented by kind of the red-orange, and then the larger myosin filaments are the kind of purple ones. And when you contract that muscle, you're pulling from both the left and right, and they ratchet together, and this entire muscle fiber shortens, and that is the contraction. This actin and myosin contraction is not only present in muscle cells of large animals, it's also present in very, very tiny creatures and even within certain single cells alone. Think, for example, about an amoeba. An amoeba is this single-celled, soft-body, microscopic creature. How does it move around without muscles and without a skeleton? Well, it also uses actin and myosin to move itself around. It uses what's called pseudopodia. Pseudopodia is a word that means false feet or fake feet, basically. And so it uses actin and myosin to move itself throughout its environment. So here you have an amoeba, and let's say it wants to move toward the upper right corner. Well, it will send out a pseudopodia, a false foot, and along that pseudopodia there will be actin and myosin filaments that will ratchet together and will pull material along, and then the rest of its body will tend to flow into that direction as well. So even small little creatures like an amoeba, they use this actin and myosin, they use these microfilaments to move themselves around. Another type of motion that you see using this actin and myosin, using these microfilaments, is called cytoplasmic streaming. And you see this really well in certain plant cells. If you've ever done a lab where you took some aquatic water plant cells and you did a thin slice of the leaf and you put them on a slide, you may have seen this movement called cytoplasmic streaming. There's a water plant that you often see used in aquariums called Elodea, or some people pronounce it Elodia, and it has very, very thin leaves that make for an excellent microscope slide sample. You can stick a leaf underneath the microscope, and you can actually see the cells very clearly, and you can see the chloroplasts within the cells. Here's an example right here. Oftentimes what you see is you'll see chloroplasts that are rotating around the outer edge of the cell. So they seem to move, and sometimes it's in a clockwise, sometimes in a counterclockwise fashion. But they seem to be kind of like following in a Congo line, a dance line, all around the outer edges. Well, they're actually moving around the central vacuole that you can't really see, but the green chloroplasts you can see. This movement is cytoplasmic streaming, and it involves these actin and myosin filaments. And the reason that this occurs is because it's basically a way of circulating material through the cell and making sure that nutrients and proteins and other materials are well circulated throughout the entire cell. There's a link right here if you want to go to this YouTube link. You'll see a great example. It's a live shot taken straight from a microscope, and you'll see exactly what I'm talking about. Last of the three structures we're going to look at are the intermediate filaments. We won't talk much about those other than to say that they are obviously going to be midway in size between the larger microtubules and the smaller microfilaments. They also play a support in cell shape, and they help fix organelles into their particular place. And one thing you might want to remember or think about with the intermediate filaments is they tend to be more permanent structures. The microtubules and the microfilaments, the cell will build them and break them down and move them around and build them back up again. But these intermediate filaments tend to stay in place. They tend to be anchor points for organelles and things of that nature. So they're not quite as fluid as far as the building and then tearing them down and building them back up again. Once they are put in place, they tend to stay in place. Our very last concept for this chapter is concept 6.7. Of course, in most creatures, there are, in your body, for example, there are not just billions, but trillions of cells, and all these cells have to work together, so no cell is an island unto itself. These cells are working together to form tissues, and tissues are working together to form organs, and all the way up until you got an entire human body or an entire living creature. So this last concept is about what goes on between the cells. What about the space between the cells, or how do cells communicate or interact with each other? And it says extracellular, which just means outside of the cell. Extracellular components and connections between the cells help coordinate cellular activity. So because almost never, unless you're talking about a single-celled organism, almost never are single cells working by themselves. They're working with their neighbors and partners and throughout the entire body. And they give off certain materials, they secrete materials, they take in proteins, they give off proteins, they take in a wide variety of macromolecules, and they have to coordinate all of this between their immediate neighbors. So the extracellular structures we're going to look at include probably one you're very familiar with, the cell walls of plants. Then we're going to look at something in animal cells called the extracellular matrix. And then we're going to talk about where cells meet together, the various types of intercellular junctions. So we'll start with the one that's probably most familiar, and that would be cell walls. The cell wall is kind of an extracellular structure that distinguishes the plant cells from the animal cells. Although plant cells are not the only ones that have a cell wall, there are some prokaryotes, and so your bacteria and that type of simple creature, the fungi, and there are even some protists that have cell walls. So there are some protists that are more animal-like, and there are some that are more plant-like, and the more plant-like ones may have cell walls. The cell wall is kind of like the structural support system of the plant cell. It gives it its shape. It helps protect the plant cell, helps prevent excessive water uptake or excessive loss of water. It's made up of cellulose fibers, which we kind of already talked about, cellulose being a long polysaccharide chain that we mentioned. And then within that cellulose, there's going to be other embedded polysaccharides and proteins as well. Most plant cell walls are made up of multiple layers, so you're not just talking about a single layer of material. You have usually, oftentimes, 2 to 3 different layers. You can have what's called a primary cell wall, which is relatively thin and flexible. Then you have what's called a middle lamella, which is between the primary wall of one cell and the primary wall of the neighboring cell. And then sometimes you have what's called a secondary cell wall. It's an additional layer between the plasma membrane and the primary cell wall. So depending on the plant cell you're talking about, you may have 2 to 3 layers that make up this cell wall. So if we're talking, when I was a kid or when I was in middle school and we talked about plant cell walls, I kind of thought of them as bricks or solid objects. But if that's the case, how do plant cells share material or send macromolecules from one to another? Well, they have something called plasmodesmata. Plasmodesmata are chinks in the brick wall. They're purposeful openings or channels within the brick wall that is the cell wall. So the cell walls have these openings or channels called plasmodesmata, and that's how the plant cells share material. At the top you have a nice microscope view of the various layers of the cell wall, and then down at the bottom you have a drawing that shows you got 4 or 5 different plant cells here. And so between, if you look closely, you see you have little channels that are running through between the cell walls. Those are the plasmodesmata we talked about. Those are purposeful openings that allow the cells to share material or send material from one cell to the next. We know that animal cells don't have cell walls. They do have another kind of material, the extracellular matrix. And when I think about the extracellular matrix, I think about it's kind of really a thick paste of a wide variety of materials that kind of holds all these animal cells together. So the animal cells don't have a cell wall, but they are covered in this material called extracellular matrix. And in this ECM, there is all sorts of material. There's glycoproteins, which are large macromolecules. There's collagen, which we've discussed a little bit before, proteoglycans, fibronectin, all this huge variety of like connective tissue. Collagen is a big one. As a matter of fact, collagen is about 40% of all of the protein that you would find in your human body. So they don't have a cell wall, but they have this mixture of material that binds them all together and kind of glues them and holds them all together. And within this extracellular matrix, there's different proteins that bind to the surface of the cell membranes. So you end up with this giant conglomeration of material that holds the animal cells together. So here we kind of have a drawing or a model. The tannish area at the bottom would represent the inside of the cell. So we have the cytoplasm. Here are some of these microfilaments. We said that they tend to be right underneath the plasma membrane, the cell membrane. There's your cell membrane. And then outside you have all this material. There's fluid and collagen and what we call fibronectins and proteoglycan complexes. And basically it just makes for this sticky material that connects and interweaves all the different cells and holds them together. So as we mentioned, this extracellular matrix, it's for support. It kind of offers some structural support. It's adhesion. It's basically the paste that holds all these animal cells together. It does allow for some motion and movement. It's kind of like a connective tissue. And then it also helps regulate certain activities because it is connected to the cell membrane, and so it helps control what comes in, what goes out, that type of thing. The last thing we wanted to mention in this chapter is that most cells tend to be pressed up against other cells, and so they have a lot of neighbors, and oftentimes they are in direct contact with their neighbors. So if you're looking at a liver, you've got thousands of liver cells all jammed together, and they're touching each other, and where they come into contact, you've got these different junctions. They have different ways where they are connected. So we're going to look at a couple of them. One of them we already talked about, plasmodesmata. That's these channels between the plant cell walls. And then we're going to look at what's called a tight junction, a desmosome, and a gap junction. As we mentioned, the plasmodesmata you find in the plant cells. These are like perforations or openings between the cell walls that allow water and some small molecules, materials, proteins, and RNA to pass from one cell to the next. Here we have a nice microscope view. You have two cells, one at the upper part of the picture and one in the lower part, and you have cell walls between them. But if you see these little channels or connections, those are the plasmodesmata we were talking about. The other junctions to mention include a tight junction. A tight junction, we'll look at a picture in just a second, is where you have the cell membranes of two neighboring cells pressed together, and they're pressed together in such a way that they don't allow any space between, and they don't allow any of the extracellular fluid that they're floating in to reach into that space. The desmosomes are anchoring junctures. This is more like a kind of like driving a nail between cells, or a stapling or connecting the cells together. So a desmosome is more like a spot where two cells meet and they are anchored together. And then finally you have what's called gap junctions. Gap junctions allow for cytoplasmic material to be transferred between adjacent cells. And we'll take a look. I think there's a short video for each one of these. So a quick little animation of a tight junction. Right in this space here, you see where the cells are pushed up against each other and they don't allow the external liquid to get inside that space. That's a tight junction or tight juncture. You can right click and hit play on that. Here we have those anchoring points we were talking about, the desmosomes. So here you have two different cells, and there's two different junctions right here that are kind of like anchoring points that hold those two cells tightly together. You can hit right click and play. And then finally we have a gap junction. And a gap junction is, again, you have a cell on the left, a cell on the right, and you have these channels through here that are purposefully allowing material to flow from one cell to the next. You can hit right click and hit play. So here we have one last little model kind of showing all those different cell junctions and animal tissues. So you got all these different animal cells pressed together, and where they meet, you have these different junctions. It can be a tight junction like you see here. It can be a desmosome, which is kind of like an anchoring junction like you see here. Or it can be a gap junction where there is purposeful space that allows material to flow, almost like a tube or a straw that allows material to flow from one cell to the next. All right, let's wrap it up. So I like this chapter because I am more familiar with this chapter, and I'm hoping that you guys are more familiar with this material as well. I think it's amazing that every one of your cells in your body has all these different parts, and they all have to work together. And the interesting thing is you are made up of trillions of cells, and all those trillions of cells have to work together in a systematic way for you to be alive. But if we were to zoom in just on one single cell, that alone is amazing. So for example, right now my voice is a little creaky and croaky because my son gave me his cold, and that's probably a virus. I doubt it's a bacteria. It's probably a viral infection. But if we were to zoom in on my body, if we were to zoom down into my nose or down into my throat, I guarantee you this is a scene that we would find. Here's a colorized SEM of a macrophage. This big brown blob is a macrophage. It's a type of white blood cell, and he is doing battle. He is kicking butt and taking names. He is taking on a whole little army, a whole little gang of these bacteria cells right here. So the yellow rod-shaped cells that you see are bacteria that has invaded the body, and here is a white blood cell, a macrophage, that is attacking and will actually ingest these bacteria cells. So remember we talked about phagocytosis, where he will ingest the bacteria, that will become a food vacuole, and then inside his cell body, a lysosome will connect with that food vacuole and release enzymes that will end up destroying that bacteria, basically digesting that bacteria. And it's pretty amazing to think that this is a single cell inside my body, and this is going on right at this very moment. And for that to work, all of those cell organelles have to do their job and work together. And then to imagine that this times trillions is what it takes for my full body to actually work and do its job. Pretty amazing stuff. So the last part says that a cell is a living unit greater than the sum of its parts, and that's really true. Cellular function arises from cellular order, and the cell and what it does is a function of that order. So let's wrap it up. We do have a summary at the very end of the chapter. The textbook always does a great job with a summary. So here's a quick summary of all these organelles that we've looked at. We started out by looking at the nucleus and the material surrounding the nucleus, and the endoplasmic reticulum and the ribosomes, which are the protein manufacturing units. Then we went to the endoplasmic reticulum, smooth and rough, which is attached to the nuclear envelope. Then we went to the Golgi apparatus, which is like the UPS of the cell, does all the packing and shipping, receives vesicles on one end, sends them out on the other. Here we have the lysosomes, kind of like the trash truck of the cell. It can recycle cell materials or digest other materials. Vacuole, large vacuoles found in plants, but vacuoles of different kinds are also found in animal cells. The mitochondria, the power plant of the cell, both in plant and animal cells. Mitochondria, it is thought that perhaps was its own cell that was taken in and became part of the eukaryotic cell. Chloroplasts only found in the plants or the plant-like creatures, and these also were thought to perhaps at one time been their own cell. Both of these have their own DNA material, their own ribosomes, and they somewhat work independently of the rest of the cell. Chloroplasts found only in plants, perhaps once again as part of the evolution of the eukaryotic cell, were taken in at some point. Then we have the peroxisomes that we mentioned. These are the ones that we don't really have as much information on. Some scientists think that perhaps, again, they could be a third candidate for that endosymbiotic relationship, but other scientists disagree. Peroxisomes create hydrogen peroxide, and they have some interesting jobs that they do. It doesn't really give us a summary for the skeletal parts of the cell, the cytoskeletal parts, so you'll have to look back in the textbook for that. I hope you feel more confident about this chapter. I know I feel pretty confident about this chapter, and even though I do have a cold, I think we're going to do well. Our next quiz is coming up. It will only be about this chapter, Chapter 6. So if you have any questions, see me online. Talk to you later. Bye.